We will now hear how academia and venture creation can work hand in hand for national development from a life sciences entrepreneur with a track record of developing and scaling businesses starting from concept, global operational experience, and early-stage investment acumen. He has successfully developed and commercialized over a dozen life sciences tools and medtech product lines in the US, EU, and China. Please give a warm welcome to Group President and CEO of Esco Ventures, XiangQian Lin, or XQ Lin.
Hello, everyone. Good morning. So first of all, thanks to the organizers, wonderful Dr. Giselle, for inviting me to speak today. That's great. Yeah. So I'd like to share our work in venture creation and partnering with stakeholders, especially with academic investigators, and also, leveraging the talent, from, let's say, postdocs. As we know, we have, in general, in the world now, oversupply of postdocs in the life science industry, and how we are putting together, very exciting new companies and developing and translating these technologies, overcoming the so-called valley of death, in order to create, a lot of valuable jobs and also address unmet medical needs. So this is what we refer to as our the venture creation model. So first, just a very quick background about Esco Group. We are headquartered in Singapore, and we have over 1,000 employees, and also operating in, we have operations in about 20 other countries, including here in the Philippines. We have, quite a large marketing office and also domestic sales and service, and looking to start some R&D projects here in the Philippines as well. Our original operating business is in life science and medical equipment, and we have commercialized these products and exporting them to over 100 countries. And leveraging this expertise in the life science industry, we have also expanded into medical devices for IVF, for infertility, to automate and standardize the embryology lab workflows to improve the clinical outcomes and patient satisfaction. Also pharmaceutical equipment, bioreactors and tools and relevant services. And last but not least, also Esco Ventures, which is our corporate venture arm. And in the context of this talk here, venture creation. So in the interest of time, I won't read our mission, but yeah, basically, of course, our vision is to improve human health and make the world a better place. So our current group corporate strategy is to create this ecosystem from our legacy business of the life science tools, which giving us market access in over 100 countries, to our fertility technologies, medical devices business. We also have biomarker discovery and development projects. So because of the success we had in our IVF fertility business, we decided to focus on unmet needs in the field of maternal fetal medicine, obstetrics and gynecology. So we are working on discovering and developing biomarkers for the prevention of premature birth to enable early prophylaxis. So that's our efforts in precision medicine. In biotech, we are focused more on venture creation. So I will explain our process later on, so I don't go into detail now. We also have bioprocess tools and services arm. This is focused on new bioreactor and bioprocess technologies, mainly to address unmet needs in the areas of novel modalities. So as we know, what is a drug nowadays has quite changed. Of course, not only classical small molecule or antibody, but also cell therapy, gene therapy, engineered microbes as drugs, have just entered the clinic. Even you have digital therapeutics now that are being developed, as a drug with a proper clinical development pathway, and also securing a high level of reimbursement in the United States. Last but not least, we are also launching an external VC fund platform with external investors to be focused on women's health and fertility. Okay, so for biotech, first of all, just to be clear, we refer mainly to therapeutics, to drugs. And we are focused on finding the intersection of novel biological insights, right? So we are really privileged to be living in this day and age. And we are, humanity is uncovering and teasing out at the molecular level, the pathophysiology of disease. So we are now able to more rationally design interventional agents, drugs, to intervene specifically at certain nodes in the biological system to change the disease phenotype. So this is what we refer to as the novel biological insights, right? Secondly, we are looking into the what we refer to as the platform technologies. So we now generally have a preference for truly novel modalities. I will describe a project that we are in the process of licensing the technology from the University of the City University of Hong Kong and to start a platform technology company. So by platform technology, we generally refer to, really truly novel modalities that will be able to generate multiple drug candidates. But it is also reasonable, to put together these platform technologies based upon claims to biological insight in specific area. For example, we are recently looking into the area of complement. We have some, very nice validated targets, C5, that is already blockbuster. And of course, last but not least, we are focused on unmet medical needs. For the interest of commercial viability, there are many unmet medical needs, but we have, of course, to filter the ones which are more commercially viable and also present a shorter and faster path towards human proof of concept in the clinic. So yeah, basically we have a team, that's located mainly in Singapore, but also around the world. And we are continuously, looking for these, the intersection of these three circles. And what we do is that at the intersection, we generate what we refer to as venture hypotheses. So this is really a hypothesis in the scientific sense of the word, but it is, it's a venture hypothesis, which would, obviously there's a scientific component, and also kind of a business or commercial viability component. And then we have a systematic process to test these hypotheses. And then, the ones which are the fittest, the ones which have the most merit, then they end up surviving. And then we would fund them as a project. The project, is scientifically oriented, to generate, data from some killer experiment that will validate or disvalidate, the scientific aspect of the venture hypothesis. And then based on which, if these killer experiment going well, then we know we have something really good on our hands, then this becomes, into a company that we will then fund and then eventually bring to the market for fundraising. Yeah. So obviously the partnership here with academia is of course the source of the novel biological insights, and also at the interface of cell therapy, kind of engineering, genetic engineering, and also of course very smart chemistry. For example, now, as some of you may be aware, we are even able, well, we are not working on this project, but there is a huge interest in, new chemistry modality like targeted protein degradation, or also, using small molecule to directly target RNA. So yeah, this kind of, clever kind of chemistry is mainly coming from academic source. So our team is mainly, spread out between Singapore, China, and the US, and we are leveraging, the synergies, from the different locations. And of course, we are looking to start some exciting projects here in the Philippines as well. So we have global team spread across these different regions. So in the interest of time, let me describe, give you some case studies for some actual projects, I think this will be maybe the most interesting. So first project that we have started is to predict premature birth in asymptomatic women early on in order to enable prophylaxis with existing agents. Existing agents would be, for example, progesterone or serclatch. So we did not find any actionable academic technology. I think Dr. Lewis gave a very nice talk just now, about, the whole developing the tech transfer, So unfortunately, in biomarker business, a lot of academic, they rush to publish and then they didn't file the IP properly. So there are some good biomarker, but we could not, actually work on them. So we started from our own, from own hypothesis. We basically did the discovery bioinformatically, and then we've completed validation in retrospective cohort together in partnership with the University of Melbourne, where they had created a very unique biobank of cervical vaginal fluid samples. So we achieved very nice results, sensitivity and specificity on the retrospective cohort. When we did the discovery, it was not one of the typical case control unbiased. We actually bioinformatically discovered the marker. We shortlisted 10, and then in the retrospective cohort we have 8 markers in a panel that shows a good that in combination has good good results. So we have just launched the earlier this year largest clinical study in the world in premature birth in Hong Kong, Australia, and China in order to collect 15,000 samples to validate these markers. And of course we're developing this into an in vitro diagnostic kit. Multiplex immunoassay, so highly deployable. Another project that we have started is, so obviously there's a trend now to go back into nature, to look for new sources of drugs. I think a lot of nice work in marine, drug discovery in the Philippines. So we know that the microbiome is clearly associated, with disease and health. And now the focus is on the metabolites of the microbiome, not on the bacterial species themselves. So, we know that breast milk is associated also with, health of the newborn. So we hypothesize, this is still a science project, that we can go into the breast milk microbiome and look into the metabolites at the genomic and metabolomic level, and then fish out, interesting new novel molecules. So this is a science project that we started internally in our lab, and hopefully we reach a proof of concept, fish out a novel molecule against existing target by next year. If not, then we shut it down. So it's a very disciplined process. So last but not least, this is academic technology we are licensing from the City University of Hong Kong. They have developed a way to engineer red blood cell extracellular vesicles as a new drug modality in itself, and to package antisense, siRNA, and also messenger RNA into these vesicles for delivery, to potentially, delivery to targeted, tissues and organs. So very exciting, new modality, and world-renowned molecular biologist Harvey Lodish will be joining us as a scientific co-founder in this company. So we are very excited, and we will be hosting him in January. And this is, a good example of, academic technology, publicly funded research that, now being translated into biotech startup, to develop new drug modality. Yeah. So thank you very much for your time and attention.
Thank you very much, XiangQian, for sharing your exciting developments in this area of